Hello there, everyone, and welcome. This is Professor Thayer here with you today to talk about my research. Now, I have a talk coming up at Yale University, so this is going to be my practice talk for that engagement. So let me go ahead and share my screen. I don't have a share button for some reason. There we go. Share screen. All right, there we are. And here's the talk. All right, so hello, like I said, I'm Kelly Thayer. This is my talk from Network to New Drugs and Molecular Dynamics for Allosteric Design. Now, I think my audience is maybe a little bit more grand scale, large scale, organismal scale than my work is at. So I am going to be talking about molecules and some of the work that we are doing for molecular design to make a new kind of molecules. And so I thought that I would give a talk that's a little bit more high level, talking about like what are our strategies for doing this and the kinds of things that we're trying to understand, give you a bit of insight into what we have recently discovered, but also just give you a preview into how we're thinking about tackling the problem. So I will be getting into some work that is very much cutting edge and emerging as we speak. So I hope that you enjoy the talk, and thank you so much to Daniel for the invitation to be here, and just so glad to have the opportunity to speak with you all. So let's get started. Okay, that slide needs to come out. All right, so looking at the overview of our work, I just wanted to give you a sense of how all the various pieces are fitting together. Now, this is a very big, very busy slide that overviews the entire talk. So the problem that we're trying to address here is we have a protein called P53 that's involved in cancer. Okay, so if this protein gets mutated, it's the guardian of the genome. If this protein gets mutated, then it doesn't have the ability to do its guardianship job anymore. Why is it the guardian of the cell? Well, because what it does is it has the ability to bind to DNA as a transcription factor to turn on and off genes. And if it detects that there is DNA damage in the cell, it will bind to DNA either to bind to genes that do DNA repair, or it can modulate the cell cycle. We'll see some more about this coming up. But suppose it became mutated, and it doesn't do that anymore. This is how cancer can happen at the molecular level, in a very simplistic way. So what we're trying to do is think about a paradigm where you have a protein like p53 that gets mutated and somehow stops having its function. But if at the molecular level we could understand what went wrong and what it's supposed to look like, what we might try to do is engineer some small molecules that would cause it to go back to doing what it's supposed to do. Now, this has been attempted for some decade or more by Alan First's lab. Alan First is a pretty smart guy. And so if he's not able to do it, maybe it's a little crazy that we're taking this on. But what we're doing that's different than what others are doing is we have tried to employ machine learning techniques in order to do this. So you've probably heard of AI and machine learning kind of things. They're useful for stuff like ChatGPT and self-driving cars, predicting what you should buy next in Amazon and all these kinds of things. But our idea was, what if we had molecular dynamics representations of what a p53 should be, and we also knew what it was doing when it was wrong. And we took some kind of machines and we said, make it go back to this working one. How could we, by engineering a molecule that binds, This is essentially the problem that we are trying to tackle. And so let me just give you a little bit of insight on what is happening here. All right, so MD simulations of our P53 DNA system in a full water box. I'll be showing you some details of all of these steps as we go through the talk. Again, this is just the overview. We have some simulations of all of these things. We want the machines to make molecules to bind. So we have some molecules that are bound that are known experimentally. So we have recently contributed some knowledge to this field. There's information that they bind, but not molecular structural information, which is what we added to this. So we could then say, okay, so this is how well they restore. Now there's a very important study in here that goes along with all of this. This is the Y220C story, where there is a wonder drug proof of concept that shows when you bind one of these particular molecules to p53, that it in the rodent models that the tumors caused by the Y220C mutation are go away and the animals are cured. But it can't pass clinical trials. It has too many side effects. It's a cysteine covalent binder, and this is a problem. However, it does exactly what we need it to do. But the surprise was that it didn't bind at the 220 site. It actually bound at a different site, so it is working allosterically. So this is what got us in the business of allosteric drug design. Another piece of the puzzle. Okay, so the GANs, we have information about how to dock molecules and send that information back to the GANs. Another piece of the puzzle is long range signaling. Okay, so if this is the DNA and these are where the molecules bind to, this one is kind of close. It actually isn't touching the DNA. It's actually maybe not the best view here, but this is actually not touching the DNA. We need a way to tell the models whether the molecules that we are designing here. So these guys are over here trying to synthesize molecules. They're using a graph. Right now, they're using graph theory, where we represent the molecules as a growing graph that is using grammar. So like you would construct a sentence by putting words together. So too, you can grow molecules using certain rules about how to put molecules together, and we can do this. So they are making molecules, but of course they need to bind. And so we can do docking studies in order to do the binding. And like I said, we have some binding information, but just because something binds doesn't mean it's actually going to interact with the DNA in the appropriate way. So to get those long range directions, we have these parts over here. These are the construction of the electrostatic networks. So the electrostatic networks are constructed from the molecular dynamics simulations. So when you do MD simulations, there are rules of physics in there, and those rules of physics involve multiple parts, and one of those parts is the electrostatic interaction, that is like charges and so on. And so what we could do is we could use the electrostatic networks. That is the interaction of the charge component, which we chose because that seems to be because in our earlier studies, that was the part that was the most telling for what was going on here. I'll give you some other reasons too coming up why we picked electrostatic. We would have our electrostatic networks. And once we have the networks, we could do some random walks inside the network to see whether there is an electrostatic signal being sent through the network down to the DNA. And so this information is also needed by the GANs because they need to do a bind and they need to do the long-range electrostatic interaction network in order to find out whether or not the molecules they're making are any good. So this is what we're trying to do. This is a very good example of declarative programming. That is the idea when we have in mind what we want to happen, but we don't know exactly how to do it. This is precisely the kind of a problem we have. So we have these pieces from the MD simulations from which we make the electrostatic networks, and then we can have the random walks in those networks, which is linked with the AutoDock-Vena docking. Because we need to dock the compounds, then we will attempt to have the compounds scored up. The GANs need improvements to their scoring functions, and we are sending feedback now from multiple, so we're at the point now where these exist. We are considering whether GANs will be the only way to do this. There are other options of models of how we could do the training. GANs is what we've explored first. And we are providing some feedback, but we are in the process of overcoming some of the challenges that are needed in order to provide this information back to the machines to continue that process. So I'll tell you about all of these things. All right, so part one of the talk will be about capturing allostery in P53 to begin with. What are the results we have from the MD simulations that make us think that any of this is even feasible or possible? So here is just a very simplified worldview of what allostery is. So just so we're all on the same page, in the classic allostery, you have some allosteric protein where you have some binding site where you want to bind to a ligand. So let's say we want to bind to this green ligand here, but the problem is the binding site is occluded. So we might do something such as bind another ligand, an allosterically active ligand at a different site than the ligand, which is far away, that causes some kind of a conformational change in the protein that then makes this binding competent. Now, the issue could be that, so it may not be a physical change. It may instead be a change in the energetic landscape, but we'll represent that as a physical change here for right now. And of course, the big question is, how does that allosteric signal get across? And this is what we are thinking about. Okay, so let me ask you to stretch your imaginations a little bit and consider a mutation as allostery. It's not typically how we think about it, but let's consider how this might work. So if you have a wild-type protein that binds to some ligand and then some mutation happened, we may now have an occlusion or a change in the energy landscape that means that it can no longer bind to its desired ligand. This is like when we get a mutation in a protein. But we might have some molecule that we could bind in addition, so you have this sort of a double allostery, if you will, where the binding of this molecule somehow reaches across and causes this to now be binding competent, which then allows it to bind to its ligand, the green one, again. This is kind of what is going on in our situation. Imagine a P53 that's happily working, it acquires a mutation, and then some molecule, maybe that wonder drug PK11,000, binds, and now it's back to doing its normal activity. Well, as I mentioned, we can't use that one though, so we need to think about how do we design this other molecule? And so if we do bring in our GANs or other machine learning machinery to do this, we're going to need to have a network representation of this because GANs can work with those. They know how to manipulate these. So what we might do is, if you will, go from here, a protein, as we might think about it, in the structural level, and convert this into a mutational network. So the network may have some nodes on it, and these nodes might represent, for example, residues in the protein. We could even do atomic level, but right now we're doing this level. So in such a case, we may acquire a mutation, right, that then somehow changes the network and prevents it from binding, and then the same similar story. But the useful thing about the network representation is that the allosteric signaling then may have something to do with how the information is traversing the network in ways that would change how the interaction happens at the other end. And so if we could cast the problem as a network, this would make it more readily worked with by our machine learning models. So that is the basic idea of what we're thinking about. So just a little bit about p53. It is the tumor suppressor protein, the guardian of the genome, as I had mentioned. The crystal structure of this was solved back in 1994, but that is only the DNA binding domain. p53 has a number of structural loops that are important. You can see how they're reaching very close to the DNA down at this end. It also has a zinc that's very important, but this is only the DNA binding domain. The full-length p53, which we have further back, even with the PK11,000 bound to it, is much larger. And yeah, here you can see how this is not on the DNA. So we have recently actually written some programs. We have just submitted a paper. It's actually under review right now. We just sent in the revisions to this full-length p53 paper where we have studied the full length. As it turns out, p53 actually exists in a variety of isoforms, for which these are the two extremes. Isoforms means that there are normal variations in proteins in our bodies because of the splice sites in the messenger RNA. So different cells may actually splice it differently, and these are two extremes. This is the easier one to study. This one is an intrinsically disordered protein. We have these N and C terminal domains attached to the DNA binding domain. Green is DNA binding. These N and C terminal domains are shown here, which account for half of the protein, which oftentimes gets ignored. So I'll be telling you a little bit about that as well. So if we wanted to understand the allosteric restoration, I wanted to show you a little bit about the Y220C wonder drug. I mean, the PK1000 wonder drug here. So here's the Y220C mutation, which is clearly not near the DNA binding site. And here's the PK11,000. Again, when you look at it in this view, it does look a little close to the DNA, but if you just poke here, you see it's actually above it here. And so what we see is that we have two situations of allosteric interaction with this system. So we need to somehow figure out how this would work. So although the Y220C and PK11,000 structures were determined already, there wasn't actually a mechanism known of how it did allosteric restoration. So we wanted to shed some light on that by doing some MD simulations and analysis, which we have done on the DNA binding domain, this one, and also on the full length. A little bit about the PK11,000. It binds covalently to the residue 182, and it improves the thermostability of p53. It also helps the protein to refold. So there are some antibodies that are recognizing the 3D structure of this, and they are confirming that the 3D structure is correct. And it also has the ability to restore several p53 dependent transcription of target genes. So we have a nice mix of rescue ones and also repair enzymes in here. This is going to be able to do both of those. And also this tumor cell death in the humanized mouse model. So I mentioned about the animal models. They've been engineered to have the human sequence instead of the murine sequence, and the tumors go away, which all suggests that PK11,000 is working to restore p53 activity. However, what wasn't known is how it actually did that. So we ran molecular dynamics simulations on this and reported some work on the structural information about this now about 3 years ago. So what we did is we looked at the dynamic ensemble and did some cluster analysis. We arrived that 2 was the best number of clusters, and we see that we have the wild type and the PK11,000 bound one are clustering together, whereas the mutant one mostly clusters outside of this. And here are the centroids over here superimposed on top of each other, suggesting that there are some differences. Now, they are subtle, as you can see. Nevertheless, there are those. So what we wanted to do then is get down into more of the details of how this actually propagates out. Like, how is this in fact actually different? And so in order to get at this, we wanted to take a look at the hydrogen bond analysis. So this is one of the things we ran. This is what the wild type looks like when it's interacting with the DNA. So these are the amino acid residues, and I just have some lines showing the interactions with the places where they're interacting. Some of them are interacting in sequence-specific ways because they're reading out the DNA major groove, which has pluses. It has basically hydrogen bond donor and acceptors in a pattern that is non-identical, right? So each of these bases has a different pattern, and so they can recognize that pattern. Others are interacting with the backbones. So let's take a look across wild type PK11,000. So when I say PK11,000, I also mean that it's the mutation is present and the Y220C. So the outlier is the Y220C because look at how many hydrogen bonds it has. It has way more hydrogen bonds than it's supposed to. And the PK11,000 is much closer to the wild type in terms of the hydrogen bond pattern. And so our understanding of how this works is that the long-range allosteric network is causing changes in the hydrogen bond, how it actually interacts with the DNA. Furthermore, we wanted to understand what was happening with the number of the small molecule binders. So there have been some follow-up to the PK11,000 in the literature. So these are collaborating groups and so on had put out a number of binders. Actually, now there's another study in addition to this one. But at the time we did this study, there was a subset of 27. We're now up to 66. But there was no information on where they actually bound to the molecule because no structural information had been determined. So our contribution then was to go ahead and do a docking study to see where they go, which we completed. We see that a number of these are docking in a variety of places. This right here actually is factor 8. So this right here, this section, is more or less almost a continuous cavity that we are using for some of our other studies because there are a couple of molecules that fit in here. So in this study, we figured out where they best dock to using AutoDock-Vina, and we also took their binding affinity and were able to show how they worked. So the ones that were the top scoring, we did go ahead and run some MD simulations to gain insight into how these were working and compare and contrast that with how PK11,000 was working, and they were not identical, but similar is the bottom line. From there, we also did another study. This is a embedding error analysis, and the reason why we switched over to this is because the superposition of conformations is a little bit problematic because this basically the things that are in the center of mass are going to be closer to each other, and the things that are just happen to be further away from the center of mass will artificially have a different distance. So one needs to somehow account for this. So one thing we could do is we could have non-Euclidean measures, and we had also been exploring these embedding error analyses. So I'm going to show you how we did this. So the first thing one does is they get the simulations, which I already mentioned we had. We'll have the wild type, mutant, and PK11,000 rescued, and then we're going to study these other ones from the second study. Then we can compute the pairwise electrostatic interaction network using some already existing well-established software called Amber for molecular dynamics simulation. We then can proceed to do the heat kernel analysis. So the heat kernel analysis will give you the flow of heat through the network. This is a way of studying the network properties of this, and the heat kernel analysis will transform the heat flow into a latent space, which is the embedding process. So in other words, instead of working in Euclidean space, this will act as a transformation of a tensor to work in an alternative space that is representing flow of heat through a network. So of course we don't actually have heat, but we do have a signal. But you could plug the information in and it will behave just as though it were heat. So then we will apply the Wasserstein distance to compare the embeddings between a test state and a reference state, and the test constructs will be our familiar PK11,000 and Y220C mutant, and we also have the DNA, the binding DB compounds that we mentioned, and we might also eventually do some studies with GAN-generated compounds, right? So anyway, one could dump in whatever they wish, and then we could use this to determine whether a test contract is similar or not. So what we did at this point is we did a comparison between the various compounds from that study that I mentioned. So these colors match up to the labels that are presented here. So what we have is the residues 96 to 290. This is the DNA binding domain, and we computed the embedding error. So what one does then is you compute the difference in the embedding error. It's called embedding error. It's not really error in the sense that it's wrong. It means how the magnitude of this that is being used, how different is it from your reference state, which we used as the wild type. That's why it doesn't appear in these graphs. So these are all computed with respect to the wild type. And what we get out is that the average embedding error summed over the errors that are shown here in this graph are ranked just like so. What we get is that Y220C is the most different from the reference state of the wild type, whereas PK11,000 is the most similar. That is, it's almost rescued according to the signal. And so we have also rank ordered these compounds that we also tested. So we have these 4 new compounds for which we computed the simulations, as I had mentioned. Here's a network that we use to do this, and this is the information structurally mapped onto the P53. So having this kind of information, what next thing we wanted to do is we wanted to move over into the into making some generative models. How do they even work, and how are we doing so far on the molecules? So this is gonna be an introduction to generative adversarial neural nets. We are also thinking about some other models. We're thinking about diffusion models as well. But let me show you how this works. So to go from so the building blocks of all of these models are really just the perceptrons. These are really quite simple. You have some kind of a black box or a white circle that is somehow making some decisions in here. And then you have some inputs. So here's your inputs right here. We have 3 here, and then you will get 1 output. So in this case, we might have a perceptron that has a very simple output algorithm. So it could be a binary one, like 0 to 1, and you would have these some weights associated with each of your inputs, and you would sum over these, and you would ask the question whether it's above or below the threshold, and this would give you your binary output. Now, this is a tunable thing, so what you can do is during the training, you would have some known outcomes, and you would tune your weights. So this is when you're doing your training, this is what is going to, in fact, actually basically works like a fudge factor, right? You say, well, what weights will this be? You could also adjust your threshold to say, okay, what will the weights be and what will my threshold be to give me the known pattern? And then once you have established some credibility on it associated with some metrics. So if you do this on a known system, you would know like how many it got wrong. And so what you can do is you can then put these together. You can have input layers, right? So here are the first ones. So you have multiples of these, and then they're providing information to the next layer, and so on till you get to the output layer. Now we also have this thing called the hidden layer. Now the hidden layer, what does it mean? Is it you can't see it? Not exactly. What we mean by hidden is that it's not the input or the output. So it's something that happens in the intermediate. And so these things are going to be represented in this abstract way. So we also don't know exactly what it is. So it's also hidden in the sense that we don't, as humans, exactly know what the meaning of those perceptrons is going to be. So to train these, it's basically a minimization problem. So just like we would have in molecular dynamics simulations, early steps for energy minimization or many other things, there are different models we could look at. So for example, a gradient descent. So we might say, okay, how can I minimize the error that I'm getting out? So here we're going to have some learning rate and number of training steps, and we're going to look at basically at the slope of this. This is going to be your del of the Laplacian. So essentially what you're looking at is how can I make downhill steps? Oftentimes we want to also allow temporary uphill steps. So you see there's a little bit of this in order to test out if we can get into a global minimum. So again, we have the simple output. This is exemplified for that, where we would have simple tunable parameters where you're optimizing your Laplacian in order to figure this out. And that is how you would get to your minimum. So what about adversarial neural networks? Okay, so adversarial neural networks are basically going to be two networks being trained at the same time. So the way this is often explained is there's an actor and there's a critic. So we have this actor robot over here that is learning to make artwork, and looks like this robot is in kindergarten and making these cute pictures. Very nice. However, it's not really quite holding up to the standards of the Mona Lisa or these other Van Gogh and so on in the real art museum. So but we might also have a critic that is going to be telling the answers to whether the artwork is any good. And the issue is that in the beginning, the critic also will not really know very well about what good artwork is. So this is why we need to send them to the art museum, maybe for some lessons for both of them so that they would both be able to learn how to do this. And so as the actor gets better and better at making pictures that look more and more realistic, almost like the real thing, the critic is getting better and better at deciding whether it's a counterfeit or not. And so as this process unfolds, what happens is the actor gets so good at counterfeiting that it actually is as good as the real thing. And the critic then by pushing the pressure on with the critic, is able to move this into a better and better form. And so the product would be, it's so good that it actually learned how to paint and would be able to make useful ones. Of course, in our situation, we're not interested in making paintings, but instead in making molecules. So we also have this reinforcement environment. So the idea in reinforcement is that you know what you want to happen, but you don't actually know how to get there. So it's kind of like video games. There is a situation that is you find yourself in, and you have to just like start making moves to figure out what to do. So first of all, you'll start off with no points, and you say, oh, there's a fire in the room, and there happens to be a faucet running over here, and you say, okay, how can I get points? So there needs to be an environment, so you make the policy, and that is something that provides points if something good happens. So for example, if the robot were to come over and touch the fire, that would be a bad thing, and so we realized not to do that. But there might be like some partial points if we go over to the water. And it might get even better, super even, by putting water onto the fire. And so the model basically learns like what steps should it take in order to improve the score. And so in our case, where we're going with this is we're trying to make p53 look more like its normal self, even when it has the mutations. So if the dynamics and the long-range allosteric signaling go back to what it used to be in the wild type. That's what we're looking for, but we don't, like in this situation, we have some ideas as to what we would do. In our situation, of course, we don't actually know what needs to be done. This is imperative programming, by the way. So we also need some things. We need a chemistry environment. So there's the size, the synthetic accessibility. So this means eventually some organic chemists will need to make these for us. So we need to be sure that there are things that could actually be made. We might want to check the solubility of them, because if they're not soluble, then that's going to have absorption issues. QED, this is the quantitative drug likeness estimate. So there are some heuristics about how they should look. There's another synthesizability library available on the size reward, and then docking. So the docking is about putting the small molecule into the receptor. Of course, this is very important. This is, in fact, actually one of the most important things that we have available to us, and it is the only one out of all of these that has anything to do with p53. The rest are about generic chemistry. So about 3 years ago now, we were able to make a model that, in fact, actually generates some molecules, and we were really pretty excited. So how did we do? Well, here we have the real versus the synthetic one. So what we can see is the good things are that there is some substantial overlap in the kinds of molecules that we were making. The real ones, as judged by the Campbell database, had similar properties to the ones that were made by our synthetic molecules. So this is positive. This was a really big step for us to actually make molecules. We're certainly not the first to do this. But what was very encouraging about this is that they more or less looked like drugs, which they did not in the beginning. So this was an important step forward. So next what we wanted to do is we wanted to make this have some properties that had something to do with p53, because up to this point, essentially what we had done is we had made things that were following things like the octet rule and had appropriate solubility and so on. But that really doesn't have a whole lot to do with binding to the protein of interest, let alone dealing with the long-range allosteric signaling that I had mentioned. So what we did is we took a look at some real compounds versus synthetic compounds, and we did the docking scores. And so what we can see is that this did improve a bit, because the synthetic ones are outscoring, they're getting higher rewards than the real ones by a little bit. This is somewhat of an improvement, but this is not really where we're going to need to be at for high throughput. I would not drop $1 million on making any of these compounds just yet. But this was an important milestone again for what we were doing. So coming back to our overview picture here, let's just take stock of what we've done so far. So we have MD simulations on quite a lot of things. We have the wild type p53, we have the Y220C and PK11000 proof of concept study now completed for the full length, and also for the DNA binding domain only. So we have the extremes of the isoforms. We also have the AutoDock study where we docked 27 compounds and ran MDs of the top ones, and we can see how those work. The story is similar to PK11000, although non-identical. We had the electrostatic networks and the embedding error analysis, which I showed you a little bit about that, and the correct ranking of the compounds. And I have also shown you that we had the GANs. So we have attempted to make the molecules, and we can make reasonable molecules. But the problem is that right now, in this instant, there is not really feedback to these models from any of the interaction energies or anything besides a simple autodoc venous score. So we have to do some improvement. So the models could be made better by improving the GANs themselves by or even substituting the GANs for some other models such as the diffusion models. We also have the binding pocket specificity, which could be improved upon. Right now we're getting one docking score, but what if we had information all along the pocket? And finally, we also have long-range allosteric interactions. So what about that? We have not had anything so far that provides any feedback yet. So let's talk about our progress in these areas. So these are the three areas that we're currently working on developing, and so much of this is highly experimental. All right, so the first thing we're doing is testing of the rewards functions. So again, we have this idea about the iterative refinement. So you have the agent, that is like the robot, and the environment. So there are rules about what is making things better or worse. And we would have some rewards that might be associated with taking some actions to see whether we have improved the situation or not. So in this situation, what we wanted to do is we wanted to actually take a look at a number of molecules. Here we have the binding affinity scores and we have molecules. I mentioned we had 27. This study was conducted in not 2004, 2024. And what we saw is that well, we could rank order them according to the binding affinity, which is available in my Gilson's database. So what we wanted to do is we wanted to rank order how they would be doing according to our models when we rank order them according to their binding affinities. And we see that more or less the trend is holding, and this is just showing our top molecules. So what we want to do is test the rewards function. So we could take a look at the size rewards, synthesizability, Lipinski. This is a heuristics that is used in the drug design world. We also looked at the QED rewards and the log P rewards and the autodoc vena rewards. So we're breaking it out into the individual parts that we're using. And so what we did is by rank ordering the compounds, as I showed you in the previous slide, so that the ones on the left are most like PK11,000, the ones on the right are the least like it. Now, among friends, and if you squint at this, you would probably look at this and say, well, you know, I'm not really seeing the trend, right? So the trend we would expect is that there would be some difference between the left half and the right half of the graph. And the only one where you might possibly, if you were being really kind to me, say that you could pick that up would probably be the autodoc vena reward going from smaller numbers to larger numbers. But all the others either don't show variation, or if they do show variation, it's definitely not in a trend from left to right. So it's not that surprising that our molecules may not be active for p53. However, what we can see is that they are turning out molecule-like things. So here's just a couple of them. I mean, they follow chemistry rules and so on, so that's good. But we need to do some improvements, and to do that, we took a look at the interaction energies. So in order to get the interaction energies, we are doing the docking, just like we did with autodoc vena. So we could dock some compounds. So for example, we could dock the Gilson compounds. This is exactly what we did with those. PK11,000 is a covalent binder, so docking doesn't apply. But a number of the compounds that we're turning out from Jeremy that I just showed you before, those compounds, well, we might want to know how they interact. So step one is we dock them. But the problem with the docking score that we're getting back is that the docking isn't quantitative. So docking scores usually are reliable in being able to rank order things approximately. So like your millionth molecule probably is in fact worse than your number one molecule, but if you want to argue between like places, 5 and 7, you really can't do that. And so this is part of why docking is good as a screening tool, but it cannot be used very precisely because it's semi-quantitative, I would say. This particular docking is using the united atom force field, which means you don't have resolution down to individual atoms. There's no water in there, and there is no position-specific information that is given out. And by that, what I mean is that AutoDocVina will report back only one number, and the effort required to get it to actually tell more would essentially require us to rewrite all of AutoDocVina. And so at that point, we said there could be other things that we could do. So the amber force field will in fact already tell you this kind of information. And so basically what we're doing is we're using AutoDocVina to dock the molecule, and then we're switching over into amber. This slide here represents a substantial amount of work, probably a good 18 months worth of work for doing just a few steps. So getting the parameters all work for the amber force field was quite an endeavor. This is Conrad's work, and he got that working. So we need to use the generalized amber force field, which has the parameters for the molecules in order to do the docking in amber. So swept out a distance around the center of mass of the molecules. So a water molecule is something like 3 angstroms across. You could sweep this out, so you get a diameter of 6. And we could ask what residues are there. So that's, you like basically sweep this out and say, oh, let's pull out the atomic level resolution of information from inside that shell. And then we could compute the interaction energies using CPP-TRAG, which is the module of the AMBER program, and get the interactions in a way for which we know an exact force field. We can solvate the system. We can know it all the way down to atomic level interactions. And so there really is not anything. So we're currently adding them together so that you would have at a residue by residue level, but you could also do it at the atomic level if you wanted. And so our idea then is to provide position-specific information back to our generative AI instead of giving back only just one number. So we could say, okay, at this place, like this is interacting well, or it's bad, or this functional group is not so good. And so you could have position-specific information that then the model would be able to use. So just to give you a sense of how well this is actually working, these are some interaction energies. Now these are represented as smiles. This is basically a flattened representation of the molecules. So here we have two different molecules. We have one molecule interacting, and we're looking at the electrostatic energy interaction and also the van der Waals interaction energy between that molecule and the residues that were swept out when we looked in the vicinity or in the neighborhood to see what those were, and we can see which residues it was interacting with and the extent to which they were interacting. So that is one molecule. So what might actually be useful is to look at a couple of them. So here's some known interactors. This is effector 20 and 22 taken from the Han et al. Bound study. And so the key things that we want to take away from this are that molecules do in fact have signatures. So you have a good electrostatic interaction or a bad one. So you see that this one in particular has this poor interaction with an arginine. So arginine is positively charged. So you might imagine this might have some kind of a clash or something going on here. Which would explain that. These are the van der Waals, so this would be more about steric occlusion. So having a high number would be bad and a low number would be good. But the idea here is that there are different parts of these molecules, different interactions that are going to be signatures. So some of these are better than others. So in the electrostatic realm, we could see that effector 22 is superior to effector 20 in this, and that there are definitely these patterns here that are different. So what is it, what functional groups align here that are working better? So it gives us some pointers in terms of where the problems actually are, rather than saying something about the entire molecule when you don't actually know like which part of the molecule you should edit or revise. So our next challenge is to actually get that information fed back to the molecules, which is requiring us to revamp how we're actually doing the grammars, because right now they're not set up to actually take any inputs from outside. So we are currently coding on that. All right. Then we also have the electrostatic networks. So up to this point, we really haven't said anything about providing feedback to the GANs about long-range interactions. And so we think the way to do this is using our electrostatic networks. So let's see some results and some work that is being done on this. Okay, this also was done in 2024, not 2004, sorry. So here is our neighborhood. Here is our interaction energy. So what we do is we run the simulation and we get out the interaction energy network, and that is being shown here. So it's mapped by nodes and edges, where the nodes are indicated by the numbers. Those are the residue numbers, and the edges are going to be the interactions. This is a full network so that every node can interact with every other node, but of course many are not interacting at the same level, maybe because they're far away from each other or just because the magnitude of the interaction is smaller. So this is obviously a trimmed representation, although many are left here, and the darker the lines, the stronger the interaction. This is actually the heat map representation of this same thing. And so right along the line, right along the diagonal, of course, you do get these very high interactions, and that's because by being bonded to the atom next door, you will get a lot of interaction between them. But the interesting ones, for example, are these outliers. We have high interaction energies for things that are not directly next to each other. And all of that then is quantitatively embedded inside of an allosteric network. So what we're doing then is moving forward to using such information in order to calculate a score. Now, there's a couple of different ways that we're doing this. I'm going to share this way with you right now. And so what we see here is we have a network. Now, I have suggested this network to you earlier at the beginning of the talk when we were introducing the idea of signaling in a protein. So now let's come back to this idea, but let's put some quantitative numbers on it. So imagine that we have a system where we have some nodes, right? And then we get some percentages, and these would be learned from the network. Okay, so this is a very simple version. And then we would have some pathways that are going through this like so. So what we can do is we can actually count up and quantitate what the paths through the model are going to be. So for example, I have a very simple path right here. So let's say we started here and you roll the dice. This is where the where some molecule is binding. So this will be the binding pocket. Of course, the binding pocket has many residues. This one is just showing the binding pocket is one residue, and it's showing the DNA binding as one residue. There's, of course, many more than that. But to keep things simple, just to give you an idea of how you could score the walk, you could multiply the 40% walk, right? So if we roll the dice to see where we're going to go, and we see we go here, and then we would go here, to arrive at the end, we would then have the 40 and the 50%. So you would get out a score of 0.45 by multiplying the probabilities together. So when you have joint probabilities, you do multiplication. So what one could do is do many, many different walks of this type. Now we've actually been testing this out and parameterizing this on models where we actually have more information known about the allostery in the system. So while we have some for P53, we're doing the calibration of these with the PDZ model. This is a very small protein that has much more known about its allosteric interactions. And so what we can see is that the allostery is definitely changing when the allosteric effector is on there. We still have some more work to do in this to figure out what exactly is happening in terms of the molecular mechanisms, and also in terms of like how one would do the scoring. So the way we should actually be doing the scoring is just the subset of residues that are in the binding pocket and the subset that are in the DNA binding. But what we've done so far is demonstrated that we are in fact capturing the signal. All right, here's a way that we have pathways mapped onto the P53. So this is an example of a pathway that has been constructed from the electrostatic networks in in P53. So in this case, what we can see from here is this does work in order to capture long-range signaling that happens when you have residues that are not contiguous. And so if you take a look here and you follow this along, there are some jumps in here. As we can see, there's a gray jump right there, and there's another one that happens in here. So we are able to pick up the jumps in the signaling, and there are long-range interactions that are occurring in the electrostatic environment. And so what we want to do then is use this as a proxy for whether or not long-range signaling is occurring. So again, back to the results from the Wasserstein embedding error analysis, we do believe that there is signaling that is inside of the information that we have. And so it's a matter now of hooking this up to our generative AI to provide information back to them so that they can use that as part of what is being used to score things up. So then, zooming back out, this is a fantasy of what is going on in my mind. This is actually a sketch I made over the course of about a year or so, with some little revisions and so on, thinking about how this all comes together. So there's some robots in the lobster room, which is how we call our lab, and they are making molecules that are trying to bind to P53. And so here's P53, and we've talked about a number of these different things, right? So we need to give them back some feedback, but right now they have only limited amounts of feedback. So we've done some simulations to demonstrate what's happening in P53. And then we're also taking a look at network analyses using electrostatics of how they're working. We're trying out different pieces of molecules. We've looked at different kinds of rewards. So we've improved these guys to some extent, but they're not seeing the whole picture yet. So we took a look at how we're working to improve that. So we've been doing some docking, and now we've done some calculations for the interface right in here. So this is Conrad's docking scores. And then we're also looking at the electrostatic networks as a better way without needing to do RMSD fit. So we can get out the information about whether or not they're aligning. And then we've done the random walks in the network, which we've also been showing along here. So that is how we are tackling this problem, and that is where our update is at for the present. So thanks so much for joining us. This is Thayer Lab. We are at a Mercury conference for undergrads in Yosemite last summer, 2 summers ago. These are the people who have contributed to the work. Sean, Stetson, Fernando, Conrad, Jeremy, and Ihor are the people who are at the forefront of this work. And many others are working on various other aspects of the projects, ongoing projects in our lab. So thanks very much for joining us today. And with that, we'll be happy to hear your questions. Thanks so much. Bye-bye.